We all know the feeling of scanning through a digital document, finding a typo, and quickly using a find and replace tool to fix it. It takes exactly 3 seconds and the document is perfect again. Now, zoom in past your skin and deep into your cells. Every single cell in your body contains its own biological instruction manual, your DNA, made up of 3 billion letters of code. The problem with a manual that large is scale. A single incorrect letter out of those 3 billion is enough to cause genetic conditions like sickle cell anemia. For most of human history, these genetic typos were permanent. There was no biological backspace key available to us. That changed with the discovery of CRISPR-Cas9, a technology recognized with the 2020 Nobel Prize in Chemistry because it allows us to edit the code of life. With it, a genetic disease becomes a biological typo that we can locate, target, and repair. The CRISPR system uses 2 main components. The first is a molecule called guide RNA. Scientists can program this molecule to act exactly like the Control-F search function on your keyboard, scanning the entire genome. It glides along the massive double helix until it matches its program sequence to the exact coordinates of the genetic typo and firmly locks on. The second component is a protein called Cas9. Think of this as the scissors that ride right alongside the guide RNA. Once the guide RNA verifies the exact location, the Cas9 protein cleanly snips the DNA double helix right at the source of the error. Scientists didn't invent this machine from scratch. They discovered it hiding inside bacteria, which have used this exact mechanism for billions of years as a weapon to chop up invading viruses. By hijacking that ancient microscopic war, humanity acquired a programmable, GPS-guided scissor for our own DNA. But simply cutting DNA doesn't cure a disease. It leaves a severed strand that the cell has to deal with. Fortunately, cells have a built-in emergency response. The moment a strand is cut, the cell scrambles to automatically knit the broken ends back together. Right as the cell attempts to repair itself, scientists can slip a new, corrected piece of DNA into the cut site, permanently fixing the typo. Isolating and programming this mechanism took immense dedication from a global network of researchers, notably led by the work of scientist Jennifer Doudna and her research partner, Emmanuel Charpentier. Their joint effort to map this exact biological process earned them a joint Nobel Prize. Merging the Cas9 cut with the cell's natural repair mechanism provides a complete system to find and replace individual letters in the genetic code. Today, doctors are using CRISPR in clinical trials to treat genetic blood diseases. In agriculture, researchers are editing plant genomes to create crops capable of withstanding extreme droughts and a changing climate. In medicine, specialists are using it to reprogram a patient's own immune cells, giving them the exact instructions they need to hunt down and destroy cancer. The technology is expanding rapidly. Global research centers and large institutions are dedicating entire divisions to exploring what these molecular tools can achieve. For thousands of years, science has only been capable of reading our genetic code. CRISPR has finally given us the ability to write it. If you could Control-F anything in biology, what would it be? Let us know in the comments below, and make sure to subscribe for more deep dives into the science shaping our world. Our ability to edit DNA means we now participate directly in directing the course of our own biology.